Today we're going to be looking at the action potential found in neurons. In some ways, this is a relatively simple process, given the fact that as long as you understand active and passive transport, we can easily verify and visually see what's going on in here. This is a neuron. We're looking at a very small portion of the neuron right here. So we're going to look at just one action potential, and then we'll talk briefly about its propagation down the neuron. To help us understand it, we have our membrane here. We have sodium channels. These are voltage-gated, potassium voltage-gated channels, and what are called leak channels, as well as a sodium-potassium active transport pump. This molecule is ATP, and then we have sodium molecules, which are blue, potassium, which are purple. Inside we have a voltmeter, just to kind of give us an idea of how it is. To start the entire neuron, the voltmeter starts at about -70, which is considered resting potential. Concentration of sodium is high on the outside, both sides of the cell. So potassium's concentration is high on the inside. In order for an action potential to occur, usually what happens to start is the voltage changes just a little bit, causing, usually with a signal transduction of some sort, into a pulse through neurotransmitters, things like that. This will open the sodium-gated channel. This allows some sodium to begin to come in. As that does, it increases the potential inside the membrane, mostly because sodium is a positively charged molecule. Increasing it from negative, add some positives, we get goes. As that signal continues to increase, once it hits threshold, we open all the sodium channels around this section, allowing lots of sodium to flood in. Again, facilitated diffusion, we're quickly flooding sodium into the cell, which causes the cell to depolarize. That depolarization will go until it reaches peak, at which case, at peak, the sodium gates close and the potassium voltage-gated channels open. This allows potassium to begin to leak or begin to exit. It's going through both the voltage-gated ones as well as through the leak channels. Okay. As potassium is leaving, we're decreasing or repolarizing the cell. Okay. Once it hits resting potential, not resting potential. Because of these leak channels, which are constantly allowing potassium out of the cell, versus the potassium gated channels, which only open in response to voltage specifically. Because of the leak channels, we actually have more potassium leaks out than what is needed, and so we end up actually what is called hyperpolarizing the cell. That hyperpolarization is an incredibly important point for propagation. Upon this point, the potassium gates close, and our ATP, our active transport, sodium-potassium active transport pump comes into play. We need to return the voltage back to resting. Once all the gates are closed, it does slowly begin to rise. However, we need to return it back to normal. So potassium, we need to pull the potassium out of the middle membrane and into the extracellular matrix, and the potassium back inside. Both of this would require active transport and ATP, our energy molecule. To do this, 3 sodium molecules bind to our membrane. The phosphate from the ATP breaks off, causing a conformational change of our protein, releasing the sodium on one side, allowing 2 potassiums to bind. Phosphate breaks back off, conformational change again, pulls it back in. ATP is restored through enzymes in the cell, and this process is repeated. Each time this is happening, and happens relatively quickly, we're increasing our voltage until ultimately it ends up back at rest. For our purposes, I'm just going to pull stuff in and out just to make this a little faster. But concept's the same. Okay, so let's take a second at a greater picture of the membrane and what would be referred to propagation of the action potential. Propagation simply means the movement of that action potential down the axon or the neuron. So I have here a system set up of two spots where the system you just saw is in play. You can imagine this as the nodes of Renvier. So the myelin sheath is around this area and this area. So this part is one node, this part is the next node. Again, we go through the action potential. Sodium gates open, flooding sodium into the cell, increasing to threshold, which causes depolarization, closing the sodium gates, and opening our potassium gates. Now here's the trick. The depolarization of this side, as this one starts to depolarize, but the increased voltage on this side actually causes the increase in voltage on this side, causing it to actually reach threshold. So this one hits threshold and causes the sodium gates to flood open, and our action potential continues on this part of the cell, while this one over here resets. Potassium floods out, causing a repolarization event, and our hyperpolarization, closing our events. And then our ATP, our active transport, occurs to reset the cell back to resting. All the while this one is repolarizing, this one is actually depolarizing. So as the sodium begins to flood in, we're depolarizing this cell. Okay, as this one depolarizes. Okay, but so here's the trick. Why does, as the depolarization, it gets same issue here, the higher end, the more voltage it has should depolarize it. But keep in mind that as this one is depolarizing, this one is actually hyperpolarized. As a result of that, this voltage has some influence, but not enough influence here to reach its threshold. So the hyperpolarization of our membrane actually prevents the signal from going backwards and instead maintains it moving forward. This cycle then finishes, closing our sodium gates, opening our potassium gates, releasing the potassium, repolarizing, hyperpolarizing, and then our ATP comes through and begins the process of resetting the system so that it is ready to occur again. Okay. Now keep in mind this whole process occurs in milliseconds. Like this whole section occurs in milliseconds. Think about the fact of moving from your brain to your fingers and telling them to snap. You just sent hundreds of neurons and neurotransmitters and nerve signal from your brain to your fingers in a matter of under seconds. So this process is occurring very, very quickly, but you can see the process fairly well here.